Thanks for joining today, everyone. This is our community conversation webinar about the FDA's accelerated approval pathway. So we're happy to take any questions that come up throughout the webinar. If you put them in the Q&A box, that will make it a lot easier for us to go through them. We can get started. So I'm Kellen Madden. I am FARA's Director of Patient Engagement. I have Jen Farmer on with me as well to go over accelerated approval. She's our CEO. And we also have Miriam Ray on with us, the Director of Global Initiatives, to help us answer questions at the end. And I will try to advance the slide now if it will let me do that. There we go. And thank you to our sponsors for supporting us and allowing us to put on these virtual education events. And they also support our in-person events. And we wanted to remind everyone our next event is the 2026 FA Symposium, which is a day-long event. It's on October 12th in the Philadelphia area, and you can sign up through this QR code to attend in person or virtually. And one last thank you before we start to Notre Dame's Patient Advocacy Exchange Program. We were able to work with a junior at Notre Dame, Paula Vega Rodriguez, over the summer, who spent a lot of time researching accelerated approval and putting together some of these slides that you'll see today. So thank you to Paula, and we're grateful for our continued partnership with Notre Dame's advocacy program. So today we'll talk a little bit about how drugs are approved and the accelerated approval process specifically. A big part of accelerated approval is something called a surrogate endpoint, so we'll spend some time talking about that as well. And then at the end of the webinar, we will talk about what happens when a drug is submitted for accelerated approval. So to start off talking about the drug development process, this is a process that can take several years. It starts out with discovery and preclinical development in the lab and then moves into testing a drug in humans to determine if the drug is safe and effective for treating a specific patient population. And then all of the data from clinical trials is submitted for regulatory approval, which is the final step before a drug becomes available to patients. And this is what we'll spend a lot of time talking about today. And it's important to note that although we have this image of a nice straight line across the screen, the drug development process can be thought of more as a series of different paths that a drug can take towards approval. So one drug might take a different path than another drug, and along with developing clinical trials and doing the research on a drug, pharmaceutical companies do spend a lot of time planning out the path that the drug will take towards approval and figuring out the specific steps that need to be taken to reach approval. In the United States, regulatory approval comes through the Food and Drug Administration, which evaluates and approves drugs under the statutory mandate of the Federal Food, Drug, and Cosmetic Act. And approval is based on safety and efficacy of the drug, as well as a benefit risk assessment that the FDA does to help them determine whether or not they're going to approve a drug. And to talk specifically about the accelerated approval pathway, it's helpful to compare it to traditional approval, which is the standard pathway for drug approval. And accelerated approval is a different pathway that allows earlier access to a drug for serious or life-threatening diseases with unmet medical need. One of the big ways that accelerated approval differs from traditional approval is in the evidence required for approval and the trial endpoints. For traditional approval, the evidence required is substantial evidence of clinical benefit or a measure of how a patient feels, functions, or survives. And this is typically measured through a clinical endpoint. And for FA, this could be some type of measure of neurological function or symptom progression or a slowing of progression. For accelerated approval, the evidence required for approval is something called a surrogate endpoint, and this is a measure that's likely to predict clinical benefit, but it's an indirect measure about how a patient may feel, function, or survive. A lot of surrogate endpoints are biomarkers, and again, they might predict a clinical benefit, and a good example of a biomarker or surrogate endpoint for FA is frataxin protein levels. And then the other big difference between traditional and accelerated approval is when in the drug development process, a patient is able to access a drug. For traditional approval, access only happens after a clinical benefit has been demonstrated. So after a clinical trial has measured a clinical endpoint or a measure of how a patient feels, functions, or survives. So in the example of FA, If you're talking about neurological symptoms or a neurological progression, that is something that typically has to be measured over the course of a year or 2 years. And the access would only occur after the end of that clinical trial looking at clinical benefit. For accelerated approval, earlier access occurs before the clinical benefit has been definitively proven. So to go back to the example of frataxin protein levels or changes in protein levels is something that could potentially be measured over the scale of weeks to months, when compared to a clinical endpoint, which would take more like a year to 2 years to measure. And the largest difference as well with traditional and accelerated approval is the type of study that has to happen after approval. For traditional approval, studies that happen after approval focus on understanding long-term safety through real-world data, so through patients who are accessing the drug through prescriptions from their doctors. As more patients start taking the drug for longer amounts of time, looking at the long-term safety data. But for accelerated approval, because the approval was granted based on a surrogate endpoint that was an indirect measure of how a patient feels, functions, or survives, a confirmatory trial is necessary to demonstrate that there is clinical benefit of the drug. And some other FDA programs to mention, the FDA might grant designations to a drug program that aim to expedite the drug development process, such as fast track, breakthrough therapy, and priority review designations. These designations are separate from accelerated approval, but they can be used in tandem. So for example, the FDA could decide to give a drug program fast track designation, and then the pharmaceutical company might take a traditional approval pathway or might take an accelerated approval pathway based on their specific situation in tandem with a fast track designation. And to give some history on accelerated approval and why it exists, it was established during the HIV/AIDS crisis in 1992 to allow patients earlier access to the drugs that were being developed in response to the crisis. In 1997, Congress codified accelerated approval in the FDA Modernization Act. And in 2012, it became directly apparent that accelerated approval could be really beneficial in the rare disease drug development space because the act was amended to state that the FDA should consider the rarity and unmet medical need of a condition when deciding whether they should grant accelerated approval. And because of its history, a lot of drugs that have been given accelerated approval historically have been for HIV or AIDS. A lot of cancer drugs receive accelerated approval as well as drugs for infectious disease, but several drugs for progressive and rare diseases have also received accelerated approval, including drugs for ALS, Duchenne muscular dystrophy, and Pompe disease. And to talk more about the relevance of accelerated approval in the FA drug development pipeline, currently the only drug that has been approved for FA is omaveloxolone or Skyclarys, which is approved for people 16 years and older. And this was a traditional approval based on a clinical endpoint, the modified Friedreich's Ataxia rating scale, which is a measure of neurological function. So this is the type of approval that we are familiar with at this point. But there have been several drug development programs that have stated that they are pursuing or considering pursuing accelerated approval as a pathway for their drug. And those drugs include nomlabofusp, DT216P2, and LX2006. So because some of these programs might be pursuing accelerated approval as the programs advance, we thought it was important to start talking about accelerated approval and help the community understand its relevance for FA. So I'll pass it to Jen now, who can talk about surrogate endpoints.
Thanks, Kellen. Hi, everyone. I'm glad to be here today to help get us all up to speed on accelerated approval and surrogate endpoints. As Kellen mentioned, we think this kind of approval is something we're going to be seeing more of as some of the newer treatments that are in development are working their way through the pipeline. And so want to make sure everybody's kind of aware of some of the opportunities and some of the aspects of clinical trials and regulatory approvals that might be different going forward. So I'm going to talk a little bit about surrogate endpoints. So what's a surrogate endpoint? So a surrogate endpoint is another, it's not measuring how someone feels, functions, or survives. That's the clinical endpoint. But the surrogate endpoint is measuring something that is directly linked to a clinical endpoint. And so, Kellen, if you go to the next slide, we'll give some examples of surrogate endpoints. Surrogate endpoints can be biomarkers, which might be something that we can measure in the blood that looks at a normal or abnormal biological process, or it can be what's called an intermediate clinical endpoint, which might be like a sign of the disease, something that we can measure. So, for example, in osteoporosis, a condition where you have low bone density, you can actually measure the bone density of someone. And they've shown through many studies that lower bone density is linked to higher risk for fractures and adverse outcomes for people with osteoporosis. And so, in that case, bone density is an intermediate clinical endpoint, and that can be used to evaluate drugs for osteoporosis. You don't have to wait and do a study where people get fractures and you count the number of fractures they get. As you can imagine, that would be a really long study. And as Kellen mentioned, the whole purpose of identifying these surrogate endpoints is to find out if these drugs are working in a shorter period of time, because sometimes waiting for a clinical endpoint to change could be a really long time. And so the key way accelerated approval accelerates the drug development process is by identifying surrogate endpoints that can have earlier readouts to tell us whether or not a treatment might be working. So if we go to the next slide, this is just to remind us a little bit about FA and where we might find potential surrogate endpoints for Friedreich's ataxia. So if we think about what causes FA, it's a genetic condition caused by expansion of GAA repeats that lead to frataxin deficiency. And frataxin protein deficiency, we know, causes the abnormal biology in the cell. And so thinking about frataxin protein as a surrogate endpoint in the case of FA could make a lot of sense if the treatment being developed works on increasing frataxin protein. If the treatment doesn't work on frataxin protein, but works on downstream mechanisms, maybe improving mitochondrial function, reducing oxidative stress, maybe there are other types of surrogate endpoints we could look at to try and tell us, is there improved mitochondrial function? Or in the case of FA signs and symptoms, we might look to, let's say, the heart or skeletal muscle or the brain to try and find other potential surrogate endpoints. Next slide. So this is not an exhaustive list of surrogate endpoints for FA, but just some examples. So trying to measure frataxin protein in the blood or the skin cells from FA patients could be one surrogate endpoint. But in order for us to use it as a surrogate endpoint, we have to be able to link it to some sort of clinical benefit. So is frataxin protein linked to clinical aspects of the disease? And you have to actually demonstrate that in order for it to be used as a surrogate endpoint. And I'll talk about that in a minute. When we think about the cardiomyopathy of FA, there are also some potential surrogate endpoints, and Lexeo Therapeutics has been doing some education for our community and looking at surrogate endpoints in their program. So measures of the heart itself, the left ventricular mass index is one measurement that could potentially be used as a surrogate endpoint when we can show that that left ventricular mass index and changes or decrease in that mass index is linked to improved cardiac outcome. Or another cardiac measurement people might be familiar with is ejection fraction. When cardiomyopathy has been present for a long time, ejection fraction goes down. That's how much blood's getting pumped out of the heart. And so maybe showing an improvement in ejection fraction could also be another potential surrogate endpoint in the case of FA or even other cardiomyopathies. In diabetes, there are lots of surrogate endpoints that get used: glucose, insulin. Another one is hemoglobin A1C. Hemoglobin A1C is a measurement from the blood that that indicates what somebody's glucose control has been over a longer period of time. And there are many diabetes drugs that have been approved based off of hemoglobin A1C levels. So these are just some examples. It's not an exhaustive list of the kinds of surrogate endpoints we might hear about being used in the context of FA programs. So if we think about frataxin, we have to, as I mentioned, we have to link frataxin protein levels to a clinical outcome. And first, I want to thank everyone who has participated in FA natural history studies or given us blood samples for research. This is what those samples, this is what your clinical data help us, this is how we use that data to help move treatments forward. So here, what you're looking at in this graph, along the bottom are percentages of frataxin protein measured in the blood. So starting over in the left is 0%, all the way over to the right is 100% of frataxin protein in the blood. And then going up on the y-axis is when somebody has onset of FA. And so individuals with the earliest onset, less than 7 years of age, on average have about 12% of frataxin when we measure frataxin protein in their blood, compared to, let's say, someone who has late onset FA, where they have onset after the age of 25, and their frataxin levels are higher. They are about 40% of normal, or 40% measured in the blood. And so you can see where we're trying to identify clinical predictors or clinical outcomes that could be meaningful. And so changing FA from a condition that has onset at age 7 to a condition that has onset at age 25 would have dramatic clinical outcomes. Next slide. Another way to look at this is to look at also how age of onset predicts other neurologic aspects of the disease. And so here we're looking at how many years somebody is able to maintain walking based on their age of onset. And so individuals who have onset less than 7 are only able to walk on average for about 12 years. Whereas again, the people with late onset FA are able to walk for at least 40 years after their first symptom. So they don't present until after 25, and they're able to walk for, on average, 40 years after. So you can see how we can take the frataxin levels that we measure in the blood and begin to link them to clinical outcomes that are very relevant to the disease. And this is an important part of establishing a surrogate endpoint. So this is just, again, a quick overview of the treatment pipeline. And as Kellen mentioned, there are many programs that we are now seeing in clinical development that are looking at different types of surrogate endpoints and thinking about accelerated approval. And again, like the real benefit where the acceleration and accelerated approval comes from is in the clinical trial that gets designed. We know from experience with other clinical trials, when we look at clinical endpoints like the Friedreich's Ataxia rating scale, our trials have to be done for 12 to 24 months. Whereas if we are doing a trial where we can measure frataxin protein in the blood or in skin cells, we might only have to run that trial for a few months, maybe 3 months or 6 months. And so you dramatically reduce the time it takes to conduct those clinical trials and hopefully reduce the time to getting to a potential regulatory approval and having the drug accessible to the community. Next slide, please. So one final comment about surrogate endpoints and accelerated approval. It is not one size fits all. Different therapies are going to use different surrogate endpoints based on how the drug works and how that drug interacts with the underlying biology of the disease. And so we may see different surrogate endpoints used in different trials, and also based on, how that drug is thought to benefit the FA symptoms. Is it a drug that is primarily treating the cardiomyopathy? Is it a drug that is treating maybe the neurologic symptoms, or trying to do both, treat the neurologic symptoms and the heart, but it's the drug's mechanism of action is in some way increasing frataxin protein, so you can use frataxin as a surrogate endpoint. But there won't be one size fits all. We will need a toolbox of surrogate endpoints to continue to advance therapies for FA. And so that is an ongoing active area of research for us. Kellen, if you can go to the next slide. We have a lot of research ongoing to try and develop different biomarkers, intermediate clinical outcome measures, and to be able to, provide the data that's needed to show that those surrogate endpoints are predictive of something clinically meaningful. And I think with that, I'm going to turn it back over to Kellen to come back to what happens after a drug is submitted for consideration of accelerated approval.
Thanks, Jen. All right, so once a trial has been conducted that's collected data about a drug's impact on a surrogate endpoint, all of that data is submitted to the FDA and they start reviewing the data to determine if they'll grant accelerated approval. The timeline for the review depends on whether or not the drug program has been given one of those designations that can allow for an expedited review. So programs that do have an expedited review, that timeline is a 6-month timeline. And then for those that don't have expedited review, it's a 10-month timeline for review. And the FDA is looking at a lot of different data and information while they're doing their review. So as Jen was just talking about, part of what the FDA is looking at is the evidence linking a surrogate endpoint to a clinical endpoint. So they are reviewing all of that data, all of the science and research that exists to support that the surrogate endpoint could be predicting a clinical benefit. And then they'll also look to see if the drug that was being researched in the trials did have an effect on the surrogate endpoint in the trial population. They are always reviewing safety data for the drug as well. And then they do the benefit risk assessment and they take into consideration other elements as well, such as the rarity of a disease, potential unmet medical need in the disease, and also patient preferences and patient perspectives as well. That all goes into their risk benefit assessment that helps them decide whether or not they are going to grant approval for the drug. And so I've laid out the different steps that can happen on this slide, and we can go through all of them. But again, it's important to remember that this is kind of a high-level overview, and each submission to the FDA, each drug program is going to have its own nuances. So there might be differences between one drug being submitted for accelerated approval and another drug being submitted for accelerated approval. But this is the general process of what happens. Again, a clinical trial has been completed. There's data on a surrogate endpoint, and the FDA is reviewing all of that data and deciding whether or not to grant accelerated approval. And importantly, with accelerated approval, before it can be granted, the confirmatory trial that aims to show that the surrogate endpoint does predict that there is a clinical benefit of a drug, that trial has to be in place before accelerated approval can be granted. So if the FDA does grant accelerated approval, at that point, the drug can be marketed and prescribed. So there might be some people who are accessing the drug through a prescription from their doctor, through commercial access, at the same time as there are other people entering the confirmatory trial and getting access to the drug by entering the trial. And at the end of the trial, all of the data is collected, the data on the clinical endpoint to determine if the drug does have a clinical benefit or it does benefit how a patient feels, functions, or survives. If the data shows that there is a clinical benefit, then the accelerated approval would be converted to a traditional approval, and the drug would be continued to be marketed and prescribed. However, if the data from the confirmatory trial shows that there isn't a clinical benefit and it looks like the drug does not improve how a patient feels, functions, or survives, then the drug would be removed from the market. And historically, it's important to keep in mind that about 75% of drugs that have been granted accelerated approval go on to receive traditional approval because their confirmatory trials did show a clinical benefit. And just to give some more details on the process, again, with commercial access, patients will have access to the drug while research continues to confirm that there is a clinical benefit. This would require a prescription from a doctor, and the FDA might include restrictions on the label for the drug in the same way that they would for a traditional approval. And the confirmatory trial is a really important piece of the accelerated approval process. It's necessary to confirm that the surrogate endpoint does predict a clinical benefit, so the accelerated approval can be converted to a traditional approval. Again, the trial usually has to be initiated before accelerated approval can be granted. And enrolling and continuing in the trial is really vital to ensure that the research can continue, that the clinical benefit can be proven, and that the drug could be removed from the market if for some reason the trial can't be completed or if the trial shows that there is not a clinical benefit. But again, keep in mind that historically 75% of drugs who receive accelerated approval go on to get traditional approval because they did show clinical benefit. And that was the end of the presentation today. We wanted to end with these resources from the EveryLife Foundation. They have a great video about accelerated approval, a web page, downloadable resources as well, focusing on accelerated approval in the context of rare disease drug research. So at this point, we can take any questions from the audience.
Kellen, I see one question already. So there's a question that's asking, is there a risk that a drug gets accelerated approval and it is not covered by insurance or by payers? And there's always a risk that a payer may deny, you know, paying for a certain type of treatment. And it has been in, I think, the news a bit more that some of the drugs that have gotten accelerated approval have been met with more resistance from payers because they haven't reached that bar of showing clinical benefit. But it's not across the board. It's not that all drugs that get accelerated approval are harder to get coverage for. I think it depends on, each case is unique. It probably depends on the surrogate endpoint, how well that surrogate endpoint is linked to clinical benefit, and again, the strength of the trial data. But those are things that, I think our community continues to work on in terms of, looking at data on these surrogate endpoints and clinical outcomes and getting that information published in the medical literature, and being able to educate payers about these surrogate endpoints will also be important, along with the unmet need in the context of Friedreich's ataxia. Let's see, Miriam, are there a few other questions?
Yes, we have one question wondering about the results that have came already and still coming from DT216P2 from Design Therapeutics. If this is potentially qualified, could be qualified for the FDA accelerated approval pathway using the current data with the frataxin MRI increase.
Yeah, so DT216, the way the drug works is it integrates with the DNA, and its purpose, its goal, what the drug is designed to do is increase gene expression and ultimately frataxin protein. So it makes perfect sense that frataxin protein might be a surrogate endpoint for DT216P2, and that Design Therapeutics would design their trial to be able to use frataxin as a surrogate endpoint. And I think that's why in their recent updates, they've been focused on, it tells them that it tells us all that the drug works, right? It gets in, it's increasing mRNA, it's increasing protein. But, to further design a study that can support frataxin as a surrogate endpoint would make a lot of sense for that particular treatment approach. Are there other questions? Helen, do you see
We
any?
have one question, not really related to the accelerated approval, but I think this is something we have reviewed before, and it's concerning the age of onset and how is it related to years in walking, Jen. And one person was concerned if this age of onset is in the section of 8 to 12 years, does that mean we would usually walk 15 years after that or until the age of 15? It looks like it was not very clear. Maybe we can
Ah,
go back on the slide, Kellen. This is
yeah.
slide 22. Thanks.
So I think one thing I forgot to mention is that what we see with natural history data is that the earlier the age of onset, the more rapid the progression of the neurologic symptoms. And so what this is showing is years of independent walking after the first symptoms. So after somebody shows symptoms and has onset of the disease, on average, how long are they still able to walk? Um, and so here it's for the folks who are less than 7 when they have their first symptoms, the average time that people are able to still walk is about 12 years versus someone who has onset of their first symptoms later between the ages of 15 and 24 has a longer time of being able to walk independently, about 27 years. So the earlier the age of onset, the more rapidly progressive the neurologic symptoms. And this is one way that we've been able to measure that is by looking at how long people have been able to walk after their symptoms start. Hopefully that's a little clearer.
Thanks, Jen. I don't see other questions in the Q&A, and I verified the chat. We don't have any there either.
Well, good. Well, hopefully this was helpful for everyone in just kind of giving a high level overview of accelerated approval, surrogate endpoints, and what maybe we can be expecting. We will continue to have individual webinars with each of our drug development partners who are putting, getting these novel therapies into clinical trials. And during those discussions that are specific to each therapy, we can kind of dig in a little deeper and you'll be able to kind of see what the approach is based on the drug's mechanism of action, what surrogate endpoint makes the most sense, and data from those surrogate endpoints linked to clinical outcomes. And they'll be covered kind of in more detail on a case-by-case basis, but this hopefully gives us a framework to go forward in. So Kellen, thanks for helping to organize this webinar today. Thanks to everyone who came and participated. And Miriam, thanks for your assistance as well with the Q&A. And I hope everybody enjoys the rest of summer.
Thanks all.
Bye. Thanks, everyone.